Okay, let's talk about a term that sounds, I know, incredibly complicated, linkage disequilibrium. But I promise, by the end of this, not only you are going to get it, but also you will see why it's one of the most powerful tools we have in modern genetics. Alright, let's dive in. And this right here is the best way to think about it. Seriously, forget the sciencey name for just a second and burn this phrase into your brain. A package deal. This simple idea is the key to unlock the whole thing. So, to really understand what linkage disequilibrium is, it's actually way easier to start by look what it's not. We are going to compare how genes would be passed down in a perfectly random world vs what actually happens in real life. Let's kick things off with this fundamental question. When you inherit genes from your parents, is it a complete lottery where every single trait, your hair color, your height, everything is picked one by one, totally separate from all the others, or is there something else going on? This slide just lays out the two possibilities so clearly. On the left, you've got linkage equilibrium. That's the perfect lottery I was talking about. Your gene of hair color has absolutely nothing to do with your gene for height. It's a perfect shuffle every time. But on the right, you have linkage disequilibrium, our package deal. This is where you might notice that in a population, a specific version of a hair color gene and a specific version of a height gene are almost always inherited together. They are linked, they are packaged. And that brings us to the official definition, linkage disequilibrium, or LD for short, is simply the non-random association of alleles. Now, alleles are just different versions of a gene, like the allele for blue eyes versus the one for brown eyes, and loci is just a fancy word for their address on a chromosome. So, all this is really saying is that some gene versions are found traveling together way more often than you would expect, just by pure chance. So why? Why does this happen? Why aren't all of our genes perfectly shuffled like a deck of cards every single generation? Well, the answer is physical. It's all about our DNA and how it gets passed down. Okay. So, when our bodies make sperm and egg cells, our chromosomes do this really cool thing called recombination. They literally swap pieces with each other. Think of it like shuffling. Now, if two genes are way far apart on opposite ends of a chromosome, they are almost guaranteed to get split up during the shuffle. But, and this is the absolute key, if two genes are sitting right next to each other, like neighbors, they are super unlikely to be split. They get passed on together as a single unit. Their physical closeness is what creates the package deal. Scientists can actually measure the strength of this genetic stickiness. They use this matrix called T prime and R squared. You don't need to know the math, but you need to know what they tell us. R squared is the really important one. Here, it's a measure of predictive power. Or a high R square means that if you know what one gene is, you can predict what its neighbor is with stunning accuracy. It's a game changer. To visualize linkage disequilibrium, imagine a grid of squares. Each block represents a region of the genome. Within these blocks, red squares show strong correlation, or high R square, which means alleles inherited together more often than expected. White squares show weak or no correlation, so alleles assorting independently. By grouping these squares into blocks, we can visualize patterns of genetic association across population. But, it's not just about physical distance. Evaluation plays a massive role here too. For instance, natural selection might favor a specific combination of genes that works really well together, making that package deal more common. Or you could have a population bottleneck where just a small group starts a new population concentrating whatever gene package they happen to carry. And finally, there's a mixture. That's when two separate populations mix, bringing together their own unique package deals that can take ages to shuffle apart. So, this is where the story takes a turn. Scientists had a huge realization. This genetic quirk wasn't just a curiosity, it was a key. They figured out they could turn it into an unbelievably powerful tool for mapping our entire genome, and this is how they started to see our DNA. It's not just one long, chaotic 3 billion letter string. No way, it's more like a city made up of distinct neighborhoods. These are called haplotype blocks. In between these blocks, you've got hotspots where all that genetic shuffling happens. But inside the blocks, in the cold spots, the DNA is basically locked in place, inherited as a single solid chunk for thousands of years. This means you don't have to read every single letter, you just need to figure out which neighborhood you are in. This train car analogy is the perfect way to understand the shortcut. This gives us, imagine, an entire LG block is one train car. Everybody inside is headed to the same destination. To find out where that car is going, you don't need to ask every single passenger, right? You just ask one representative, while in genetics, that representative is called a tag SNP because of that strong linkage that one tag SNP tells you what's going on in its entire neighborhood with almost perfect accuracy. Let's just take a second to absorb the sheer power of this shortcut before we understood LD. If you wanted to find a disease gene, you had to search through all 3 billion letters of our DNA. It was a monumental task. But then the International HapMap project used this strategy and realized we only needed to check about 500,000 of these tag locations to get a great map. That completely changed the game. Honestly, it was revolution. So we have the map, we have the shortcut. Now let's see how this is actually used to hunt for the genes behind really complex diseases. This is all done with a method called a genome-wide association study, or GWAS, and it's a pretty straightforward 3-step process. First, scientists take the tag SNPs from thousands of healthy people and compare them to thousands of people who have a specific disease. Second, they use powerful statistics to find a tag SNP that show up way more often in the group with the disease. It's basically guilt by association. And third, they remember that the tag SNP isn't the cause itself. It's just the signpost. It's the big flashing arrow pointing to the neighborhood where the real troublemaking gene is hiding. The hunt is on, the date is in. So, how do we read the results? Let's take a look at how a statistical signal becomes a massive medical breakthrough. This is it. This is the iconic result. The G was called a Manhattan plot, and you can totally see why it looks like a city skyline. So, the x-axis along the bottom, that's our map. All our chromosomes laid out one after another. Each dot is one of the tag SNPs we tested. And the y-axis, that's the strings of the evidence. The higher the dot, the more strongly the genetic neighborhood is associated with the disease. Anything that pokes its head above that red line is considered a statistically significant hit. But here's the key insight. This is the part that brings it all together. Why do we see these giant towers and not just single dots? The answer is linkage disequilibrium. That skyscraper isn't a single gene, it's the entire neighborhood. The real disease-causing gene is the anchor, but because all its neighbors are so tightly linked to it, they get dragged up in the state, right along with it. LD is literally what built these skyscrapers, and this is not just a theory. This is how major discoveries are made. For example, some early studies found this huge skyscraper on chromosome 16. Researchers knew that was their target. They zoomed in on that specific LD block, that one genetic neighborhood, and by testing the gene inside, they discovered the FTO gene region, a groundbreaking undeniable link to human obesity. All for a single statistical blip that pointed them in the right direction. So, think about that journey. We went from a simple observation that some traits travel in packages to understanding the forces that bind them to creating a map of our entire genome. If you found value in today's video, don't forget to subscribe, share with your colleagues, and be part of the next gen pharmacist community, where knowledge meets innovation. Thank you.